All right, let's dive into the world of GPCRs. These things are like tiny gatekeepers on our cells, controlling how we respond to, like, everything. From smelling fresh-baked cookies
Oh, yeah.
to, the adrenaline rush of a close call.
Right.
We've got some great material on the topic, and our listeners really want to, like, meticulously trace the steps of GPCR activation.
Yeah, they really want to understand that whole process.
Yeah, so let's do it.
What's so fascinating about GPCRs is they're involved in so many processes. It's not just like how we sense the world, it's how our bodies function at like the most basic level.
Okay, so let's break it down, this sort of intricate dance between GPCRs and G proteins, step by step. Like, what's the initial trigger, the thing that sets this whole thing in motion?
It starts with a ligand, a molecule carrying specific instructions. Think of ligands as keys that fit into specific locks, and the locks are those GPCRs embedded in the cell membrane.
Got it.
And these keys, they could be hormones like adrenaline, neurotransmitters like dopamine, or even light photons if we're talking about vision.
Interesting. So the ligand is the key that unlocks the GPCR. So what happens when that key goes in the lock?
The GPCR undergoes a conformational change, kind of like a lock shifting its tumblers when the right key is inserted.
Okay.
This shape-shifting is crucial because it creates a docking site for the next player in our story, the G protein.
Hold on, can we just pause for a second? I mean, it's hard to picture these changes happening at such a small scale. Like, are there any real-world examples that could help us visualize this?
Absolutely. Let's look at rhodopsin, the GPCR responsible for vision.
Okay.
It has a molecule called 11-cis-retinol bound to it, like a key sitting in the lock. When light hits our eyes, it causes that 11-cis-retinol to change shape, becoming all-trans-retinol. And that shift activates the rhodopsin receptor, which sends a signal to our brain, allowing us to see. So it's like a light switch, but on a molecular level.
Wow, that's incredible. So, like, the way we perceive the world around us is literally dependent on these tiny shape-shifting proteins.
It really is.
Okay, so we've got the ligand binding to the GPCR, causing this conformational change. What happens next in this sort of cellular relay race?
Well, that's where the G proteins, our cellular messengers, enter the picture. They're waiting on the inside of the cell, ready to receive the message from the activated GPCR.
And what do those G proteins look like? Are they just single molecules just bouncing around?
Not quite. They're actually made up of 3 subunits,
Okay.
alpha, beta, and gamma,
Okay,
working together as a team. But they don't just jump into action immediately. In their inactive state, the alpha subunit of the G protein is bound to a molecule called GDP.
Okay.
Think of GDP as like a safety switch, keeping the G protein off until the right signal comes along.
So GDP keeps the G protein on standby. So how does it get flipped on?
It's the conformational change in the GPCR that triggers the G protein to swap out GDP for another molecule, GTP.
Okay.
It's like replacing a dead battery with a fresh one, suddenly energizing the G protein.
So GDP is the go signal for the G protein. So what happens once it's energized?
The activated alpha subunit, now bound to GTP, detaches from the beta and gamma subunits. The team splits up, and each subunit can now interact with other proteins inside the cell, triggering a cascade of intracellular signals, like amplifying the initial message from the ligand, creating a chain reaction that leads to a specific cellular response.
Hold on. So one little ligand binding to a GPCR causes this whole internal chain reaction.
Yeah.
It's amazing how something so small can have such a big impact.
It is, and this cascade can have incredibly diverse effects depending on the specific G protein involved and the downstream signaling pathways activated. It can change enzyme activity, open ion channels, even influence gene expression.
That's incredible. This whole process is like a beautifully choreographed dance, with each step perfectly timed and orchestrated. But, I have to ask, like, what does this all mean for our listeners? Why should we care about these tiny molecular players and their interactions?
Well, think about it this way. Every time you smell something delicious, feel your heart race, or even just move a muscle, GPCRs and G proteins are at work behind the scenes, translating the world around us into a language our cells can understand. And understanding how they work opens up incredible possibilities, especially in the field of drug development.
That makes sense. So if we can understand how these signals work, we can potentially manipulate them to treat diseases. We've covered a lot of ground already. Where do we go from here?
Now that we've seen how this incredible cascade of signals gets initiated, let's shift gears and explore how it's controlled. Because cells need a way to turn these signals off just as efficiently as they turn them on. Otherwise, things could spiral out of control.
Okay, so how do cells hit the brakes on this GPCR signaling pathway? That's what we'll uncover in part 2 of our deep dive. Welcome back. We've been exploring the amazing world of GPCRs and how they kickstart a cascade of signals within our cells.
Right.
But as with any good party, there comes a time to, turn down the music and send everyone home.
Right.
Cells have their own ways of hitting the off switch on these signals, and that's what we're going to kind of unpack now.
You're absolutely right. Cells need to be able to regulate these signaling pathways to maintain balance and prevent overstimulation.
Right.
Otherwise, things could get out of control. Imagine if your heart kept racing even after the threat had passed.
Right.
Or your senses were constantly bombarded with overwhelming input.
Yeah, for sure. Okay, so how do cells hit the brakes on this GPCR signaling pathway?
Yeah.
What are like the key mechanisms involved?
Well, one of the main players in this shutdown process is the alpha subunit of the G protein. Remember, it's currently bound to GTP, which is like its fuel, keeping it active and allowing it to interact with other proteins.
Right, GTP is what gives the alpha subunit the energy to go off and do its thing.
Yeah.
So what happens to that fuel eventually?
The alpha subunit has a built-in timer, a special ability called GT-pace activity, and it slowly breaks down that GTP back into GDP.
So it's like the fuel's running out and the alpha subunit starts to lose its energy.
Exactly. And once that GTP is converted back to GDP, the alpha subunit can no longer interact with those downstream signaling molecules.
Okay.
It's like the party's over, and the alpha subunit heads back to rejoin the beta and gamma subunits, forming that inactive G protein complex again, ready for the next round of signaling.
It's like a cellular reunion. The team is back together, ready to respond when the next signal comes along. But I'm curious, is this GTP breakdown the only way to shut down the signaling pathway?
There's another crucial mechanism involving proteins called arrestins. They bind to activated GPCRs, sort of like putting a cap on them.
So they prevent the GPCR from activating any more G protein.
Precisely. And they also help to pull that GPCR away from the cell's surface, a process called internalization, like taking the receptor off the field for a timeout.
Interesting. So the cell has multiple ways of controlling the signaling pathway. The alpha subunit's internal timer and these arrestin proteins that can block and internalize the GPCR. It's like a multi-layered security system, ensuring that things don't get out of hand.
That's a great analogy. And these off switches aren't just passive processes either. They're finely tuned and regulated, allowing for incredibly precise control over the intensity and duration of the signal.
This regulation must be crucial for maintaining homeostasis, right?
Yeah.
Ensuring that our bodies respond appropriately to changing conditions without overreacting.
Absolutely. This intricate balance of on and off signals is essential for virtually every process in our bodies. And it highlights why understanding GPCR activation is so critical for drug development.
Okay.
We can't just focus on activating or blocking a receptor.
Right.
We need to understand the entire signaling cycle to ensure that a drug has the desired effect without causing unintended consequences.
That makes me think about those drug commercials where they list a mile-long list of potential side effects.
Exactly. Those side effects often arise from a drug interacting with multiple GPCR subtypes or disrupting the delicate balance of those on and off signals.
Right.
Designing drugs that target specific GPCR subtypes and modulate their activity with precision is a major challenge in pharmaceutical research.
It's like trying to find the perfect key for each lock without accidentally opening any doors you don't want to. But I can see why this research is so important given the sheer number of diseases linked to GPCRs. We're talking about everything from heart disease and cancer to neurological disorders and even pain.
You're absolutely right. GPCRs are involved in a vast array of diseases, and understanding how they work is key to developing new and innovative therapies. In fact, many existing drugs already target GPCRs, and the potential for future drug development in this area is immense.
So in the next part of our deep dive, we'll explore how this knowledge of GPCRs is being translated into real-world applications and groundbreaking therapies. Stay tuned. Welcome back to our deep dive into the world of GPCRs. We made it to the final part. So we've journeyed through all these steps of how they're activated, how they trigger those cascades of signals, and then how those signals are carefully regulated.
Right.
But now let's shift gears and explore how all of this knowledge is being used to develop new therapies and really change how we treat diseases.
Yeah, it's pretty amazing to think that these tiny little players have so much potential for medicine,
Yeah.
By understanding how they work, we can, potentially target them with drugs to treat all sorts of conditions.
Absolutely. So let's talk specifics. What are some examples of how these GPCR-targeted therapies are already being used? And what are some exciting possibilities for the future?
One area where they've already made a big impact is in cardiovascular disease.
Oh.
Think about beta blockers. It's a common medication used to treat high blood pressure and certain heart conditions.
Right.
Well, these drugs work by blocking specific beta-adrenergic receptors in the heart.
Yeah.
So they're basically reducing the effects of adrenaline and helping to regulate heart rate and blood pressure.
So we're already using the power of GPCRs to treat heart disease. What about other areas? Where else are these targeted therapies showing promise?
Oh, the possibilities are pretty much endless.
Wow.
GPCRs are involved in so many processes that they're potential targets for, well, everything. Pain and inflammation to neurological and psychiatric disorders, even cancer.
Wow. Can you give us some specific examples of how this research is actually, turning into new treatments?
Sure. Let's talk about pain management. Opioid drugs like morphine target a type of GPCR called the mu-opioid receptor.
Okay.
By activating this receptor, they reduce how we perceive pain. But, as we know, opioids have a lot of side effects.
Right, yeah.
Addiction, respiratory depression, things like that.
So the challenge is to find ways to, target those receptors more specifically, activate only the pathways involved in pain relief, but not the ones that cause all those unwanted side effects.
Exactly. And researchers are making, a lot of progress in that area. One approach is to develop drugs that are biased agonists.
Okay.
What that means is they can activate a GPCR in a way that sort of preferentially activates certain signaling pathways over others.
So it's like fine-tuning the signal,
Yeah.
amplifying the good stuff and minimizing the bad.
It is. And it's got huge promise for developing safer and more effective pain medications. Another exciting area is cancer treatment.
Okay.
Some GPCRs are overexpressed in certain types of cancer cells, and these receptors can actually contribute to tumor growth and spread.
So if we can block those receptors, we could potentially slow down or even stop cancer.
That's the goal, yeah. There are several GPCR targeting drugs already in clinical trials for different cancers. One approach is to block what are called chemokine receptors. These are GPCRs that are involved in immune cell trafficking.
Oh.
And by blocking these receptors on cancer cells, we could, stop them from attracting the immune cells that help them grow.
It's incredible to think that by manipulating these tiny proteins, we can have such a big impact on disease. It's like we've got this whole new toolkit.
It is, yeah. And we're just at the beginning when it comes to understanding the full potential of GPCRs. There's so much more to learn.
Absolutely. Well, it's been an amazing journey exploring the world of GPCRs, from activation to regulation to their potential for revolutionizing medicine. I hope our listener is feeling that same sense of wonder and excitement.
Me too.
About these, tiny players that have such a profound impact on our health and well-being.
And remember, the journey never really ends. There's always more to discover and learn.
That's so true. Well, thanks for joining us on The Deep Dive, and we'll see you next time.